My name is Jim Heath. I'm the president of the Institute for Systems Biology here in Seattle. And for the last several years, we've been running a large component of the National Recover Study that's funded by the NIH on long COVID. I'm the president of the ISB, and I'm also the director of the study. Our study encompasses several locations. And here in Seattle, we've got Swedish Providence. We've got UW as well. In Spokane, we've got a site, and then down south in Cedars-Sinai in LA. And so tonight we have both Helen Chu from UW, who is the clinical lead on the UW Recover study, and Jason Goldman, who's the clinical lead on the Swedish Providence, and both are co-leads of this program, as well as later on, Allie Duven from Swedish and Helen Nguyen from UW, both who are trial coordinators or study coordinators for the Recover study. So what we had hoped to do tonight is to present you with a overview of what has been happening so far. I think we are in a sort of at a apex of where the next year will reveal a lot, but we've already learned some things. And so we've got some short presentations. First from Jason Goldman. Dr. Goldman will be followed by Dr. Chu. Then I will present just a couple of slides, and then we'll open it up, and all of us will be available to answer questions. With that, I'll hand it over to Jason. Jason, take it away.
Thanks, Jim, for that introduction. Let me share my screen here and get started. So welcome to the town hall webinar tonight. Thanks for participating. I know there's a lot of patients out there in the audience and participants in our various Recover studies. So we're happy to be able to have this forum to share some of the data back with you guys, and thank you for your participation. I'm going to talk a little bit today just about updates of the Recover studies. So here's the agenda of my talk. It'll be relatively short. I'm just going to review the Recover adult cohort study. We're going to talk about the enrollment, the retention, and data collection and biospecimens. And then I'll give a couple slides of highlights of the published data, which is starting to emerge. Here's our Pacific Northwest Hub Recover study website. I'm sure many of you have accessed this over the course of the study. As Jim mentioned here, the various institutions that are participating in our hub in the Pacific Northwest. And there's lots of good resources here on this website. So if you haven't been here, I'd encourage you to check it out. It also links to the main Recover website. Here's a very brief overview of the sort of kind of complex and multifaceted nature of the Recover study. Recover has a number of different aspects to it. It has the observational cohort studies, and there's adult study, pediatric study, pregnancy study. There's also Recover clinical trials. Throughout our hub, we've been participating in these, and Dr. Chu is going to give a much more in-depth update of these. There's an EHR, or an electronic health records component, which has been very productive and has put out many manuscripts that have significantly contributed to our understanding of long COVID. There's pathobiology studies. I'll talk briefly about these. And there's an autopsy study. And then there's lots of other components of this: survey data, clinical data, imaging, mobile and digital health, and pathology specimens for the autopsy. So how have we done so far? We're a couple of years in. This slide briefly talks about the enrollment and retention of the Recover adult observational cohort study. This is data that I've swiped from the Recover portal. The total participants enrolled nationwide is close to 15,000, and the goal there was, right about there. We're at 99.1% of the target enrollment. That was a little bit reduced from what was originally proposed when some of the original Recover materials came out, but that is the full enrollment. Of those roughly 15,000, approximately 5,000 were enrolled within 30 days of their acute COVID infection. 7,000 were enrolled after they had COVID, so more than 30 days out, and you could have been enrolled up to a couple years out from your infection. And there was roughly 2,500 people who were uninfected when they were enrolled, and some of these folks have acquired SARS-CoV-2 infections on study. Retention has been quite good in this study. The statistics there are in this little figure, which is a little hard to interpret, but overall retention is about 85%, which is really fabulous for a study of this nature that's so big. These red bars indicate some people who have missed some visits, but they're not, unenrolled or withdrawn from the study, so they're still counted. So, true retention is probably somewhere between 85% and 79% in that range. And I'll show you a little bit about the huge amounts of data that have been collected from this study. There's been, as of January 1st of 2025, there's over 100,000 symptom surveys collected. So this is really an amazing accomplishment. There's also, other correlated surveys like the social determinants of health, vaccine data, pregnancy data, substance use, etc. There's been many blood tests, which many of our participants will know about as you've been receiving those results back. There's also other tests, and here are some of the, quote unquote, tier 2 tests. These are generally thought of as non-invasive tests. There's thousands of different tests of all sorts of clinical tests that assess different organs in the body that can be affected by long COVID. And some of the tier 3 tests are coming. These are things that might be a little bit more invasive, like skin or muscle biopsies that can help in some of the pathobiology studies that I'll mention. What about the biospecimens? Besides the clinical labs, Recover participants are donating lots of biospecimens. Here are some of the different types. When you donate blood, it gets separated into various blood components. Those are the top 3 there. We're doing nasal swabs, urine samples, saliva, stool, lots of different sample types. And there's, literally hundreds of thousands of these different, sample types. More than 1 million biospecimens are in the Recover Biobank. So this is a huge and phenomenal resource that's going to really help to understand and define long COVID and the mechanisms that underlie it. The NIH is funding various pathobiology studies. So far, there's been more than 60 studies funded, and these biospecimens have gone out to researchers in 12 of the studies so far. So this is an ongoing work in progress. I forgot to mention the digital health. That's also a really interesting aspect of this study. Study-wide, of the close to 15,000 participants, 45% have enrolled in the mobile health component and have a Fitbit and are tracking their health data passively, which is really interesting. There's 2 studies coming out soon about that, and that should be interesting to watch for. All right, so what about published data from Recover? So far, there have been 86 published manuscripts to date. As I mentioned, many of these are from the electronic health records cohort, and this is where the health data that's in the medical records are aggregated, and these are essentially retrospective studies, epidemiologic studies. I'm going to talk about a number of studies that come from the Recover adult cohort that I think are some of the key highlights of the information that's been generated from the study so far. And these are the 3 studies that I'll talk about: the Long COVID Research Index, a paper about standard clinical laboratory measurements in long COVID, and another very recently published paper on circulating viral antigens in long COVID. So here's the first study. This was an attempt to define long COVID based on symptoms, and it came out in the journal JAMA, Journal of the American Medical Association, in 2023. What this study did was it looked at roughly 37 symptoms that were in the symptoms questionnaires that Recover participants are filling out, and it assessed which symptoms were most common in the people who had SARS-CoV-2 infection or COVID in the past, and compared that to people who were uninfected. And the ones that were much more common commonly found in the patients with the infection, those were aggregated into this sort of score system and summed up to say who had COVID and who was indeterminate as having long COVID. And here are the symptoms. Many people on this call will be familiar with these. Feeling tired, feeling fatigued, brain fog, dizziness, gastrointestinal symptoms, heart palpitations, loss of sexual desire or ability, changes in taste or smell, feeling thirsty, chronic coughs, chest pain, and unusual movements. So after this study came out, the national academies did a really wide-ranging effort to make a comprehensive definition of long COVID. This is not part of the RECOVER study, but I bring this up because I think it set a different standard for how to define the condition. And this is really meant to be utilized in the clinic. So it's a pretty broad definition, and it starts calling the condition long COVID when it's 90 days or more out from COVID and symptoms are persisting or newly emergent. We did within RECOVER an update of the long COVID research index, and this was published just recently in 2024 in the same JAMA journal. And this kind of updated the index and also appropriately contextualized it within the National Academy's definition. So the National Academy's definition is really where we look to how to define long COVID in the clinic, whereas the long COVID research index is really useful for being able to categorize or classify people for the purposes of research study. And I like to think about this index as like defining people who are highly symptomatic with long COVID. So this figure here shows that the two indexes from 2023 and 2024 were very correlative, meaning that they tracked well together, but it just defined people slightly differently. And I think one of the main advances here is to put it into the context of the broader National Academy's definition. All right, so study number 2. This was an important paper that was published in the Annals of Internal Medicine that came out of the RECOVER adult cohort study. So in this study, over 10,000 adult participants were included, including close to 9,000 who had prior SARS-CoV-2 infection and more than 1,000 who had no prior SARS-CoV-2 infection. You can see all the different types of labs. There were 25 different routine clinical labs that were tested in the study to see which ones might be useful as a biomarker for long COVID. In the study, it was called PASC at that time, or post-acute sequela after COVID, but we're really referring to it now as long COVID. This study was a little bit thought of as a bit of a disappointment, or it's called in the medical literature a negative study. And it showed that the standard clinical labs did not distinguish long COVID. So these are some of the figures from the paper, and what you can see here is for many of the different routine lab tests, it's really quite the same. Like these lines are just horizontal, and they're really the same across the different groups. Each of these different colors is a different long COVID phenotype, or someone who doesn't have long COVID. That's in green. So even where there was very slight differences in the statistics, I mean, it's a huge study with 10,000 people. We don't really think of those differences being very clinically meaningful or not very different at all because the differences were so small. So while this type of a negative study, can feel disappointing to some, I think it is an important finding that many people with long COVID have been experiencing, that their labs have been normal and they've been told that there's nothing wrong with their labs. And we're seeing that even though people who we're defining as having long COVID in this study, their labs were not different. So I think in a way, it does sort of validate some of the experiences that people living with long COVID have been having in the healthcare system. All right. So this is the third paper. This is a really interesting paper that I think gave some real hard data on the theory that the virus might persist in the body. or be part of a reservoir that's hiding out somewhere in the body. And in this study, the researchers used blood samples from 4 different cohorts, including Recover, and looked at different viral antigens. And an antigen is something that the virus makes as it's replicating in the body. So it's not really thought to persist for a long time if there's no replication. And what we see here in this figure is that many of the antigens did persist. They were there in the beginning at 1 month, but also to 3 months or 4 to 7 months, or even longer to a year. And the figure in the middle with spike antigen showed the most positivity, with 15 to 20% of people in that 1 to 7 month range being positive for the antigen. And the other important finding from this study is that if you had an antigen detected in your blood, you were roughly twice as likely to have long COVID symptoms. So while this doesn't sort of track 1-to-1 with having long COVID, it is very useful in that it seems to be one of the first biomarkers that's been sort of validated to try to understand who might have long COVID or how we can categorize them. I think this is going to be particularly important for coming clinical trials in long COVID, and I think Dr. Chu will talk more about that. So in summary, the Recover adult cohort study has collected huge amounts of data and biospecimens. Results are amassing, and there is more to come. And I'd like to call a special thank you to our amazing participants who have donated your time, your energy, and your biospecimens to make all of this work happen. And all of this research could not happen without the participants and the engagement we've seen within our hub, but really across the whole country. It's been a really phenomenal effort. And there's my email. Please feel free to email me if you have questions, or we'll have a Q&A session at the end of this session as well. So that's all for my part today. Next up is going to be Dr. Helen Chu.
Hi, everyone. Thanks to Doctors Heath and Goldman for their talks. My name is Helen Chu. I am professor of medicine at the University of Washington, and I lead the University of Washington site for Recover. And so in my talk, I'll be going over the clinical trials that we're doing as part of the Recover Consortium. And then after that, I believe I'll go ahead and turn it back to Dr. Heath, and then we'll open it up for questions. Thank you all for joining tonight. I know that this is a really interesting time, both in terms of now having done the study for several years and having a set of data, and then also at the point at which we're really going to be able to dig deeply into some of the results and answer questions in the next 1 to 2 years. So I'm going to go over the clinical trials that have been done to date. So the first is, I just wanted to sort of lay the landscape of where we are in terms of this prior studies that have looked at the effect of vaccines and Paxlovid in reducing the risk of long COVID. And there have been several studies that have looked at these things, but I wanted to focus on 2 recent ones. And the first one is looking at the receipt of vaccines. And this was a study that was done at the Veterans Affairs Medical Center in St. Louis, Missouri. And what they did was they looked at veterans who had SARS-CoV-2 infection compared to healthy controls. And they looked at the medical records of these veterans from the period of time from 2020 to 2022. So starting early in the pandemic and all the way through Omicron. And what they found was that in these veterans, the ones who had been infected with SARS-CoV-2, that the effect of the vaccine that they received was associated with a reduction in the risk of long COVID. And overall, there are some numbers here showing that out of in the Delta era, we observed long COVID in 9.51 out of 100 person years in those who were unvaccinated, compared to 5.34 out of 100 person years in those who were vaccinated. So a lower rate of long COVID in those who had received vaccines. These numbers are slightly different when you look at the Omicron era. You can see that overall the numbers of or the incidence of long COVID was lower in those who were affected during the Omicron era compared to the Delta era. but that also during this period, getting a vaccine was associated with protection from infection. There have also been a lot of studies from Israel, from the United Kingdom, and also from the United States that have shown similar findings that really getting a vaccine is protective against acquisition of long COVID, and that this effect may have varied over time as we have entered into a new phase of COVID where most people have hybrid immunity, but that it still remains protective. The next study I want to talk about was a study that was done at Stanford. And here they looked at the effect of Paxlovid on reduction of risk of long COVID. And they did a study where they enrolled a very small population, so 63 men and 92 women, with a median age of 43 years. And what they did was they randomized them to getting either Paxlovid or placebo in a 2-to-1 manner, so twice the number of people got Paxlovid. And they evaluated them for whether or not they had any statistically significant reduction in the symptoms of long COVID. And they found that it did not have an effect. So overall, at 10 weeks, there was no difference in those who did and did not receive Paxlovid in terms of their long COVID symptoms. As many of you know, we have done a very similar study here as part of the Recover Consortium, and we don't have those results yet. This, I would say, is a very small study. It's a single site study, so it was only done in one place in the United States, and that population may not be representative of the overall population of people who have long COVID. So I don't know that we could say that this is a definitive answer to that question, and that's why there was Recover Vital, which is the Paxlovid trial as part of the Recover Consortium. So now moving on to the clinical trials that have been performed as part of the network. So there have been a few trials. There's Recover Vital, and these are the sites that participated in each of these different trials. Recover Neuro, Recover Autonomic, Energize, and Sleep. And so those are the 5 trials, and I will go over each of them in detail. So Recover Vital, I know Recover Neuro is the first trial. This one is focused on cognitive dysfunction symptoms in people with long COVID. And that includes people who have brain fog and people who have problems focusing on tasks. And so one of the ways that we wanted to evaluate this is by randomizing people to different interventions. So there was a set of interventions that we randomized people to, things like brain games or stimulation of the brain and other tasks to see what would help potentially help reduce the brain fog and the difficulties with cognition that have been associated with long COVID. So this study has just completed, and we don't have the trial results yet, but we hope to have them in the next several months. The next study that also just finished is the Recover Vital study, and this is the study looking at the effect of Paxlovid on reduction of long COVID symptoms. So in this trial, individuals were randomized to 3 groups. One is Paxlovid alone. The second was a shorter course of Paxlovid plus ritonavir, which is one component of the Paxlovid that's thought not to have an effect on SARS-CoV-2. And then the third group into ritonavir and placebo. And so these individuals who participate in this study, and I think many of you were part of the study as well, were intensively followed for their symptoms of long COVID and had multiple blood tests done as well. And we also anticipate to have the results from this trial in the next several months to share with everybody. Recover Autonomic is another trial, and this one is focused on autonomic dysfunction, so having a fast heart rate or dizziness or fatigue. And the intervention that's being evaluated is to first do a baseline visit, and then to take an active study drug and wear a fitness tracker to record heart rate and daily activity. And so this study, I believe, has either not quite started or just started, and we will probably not have results from this for at least a year or 2. Recover Energize is focused on exercise intolerance and post-exertional malaise. And so this is a study really to understand if there are interventions that you could take to help with increases in exercise tolerance and decreasing all of the fatigue that people with long COVID are experiencing. And so in this one, individuals are also participating in two groups. One is an active study, active exercise program, and the other one is a control program, and also wearing activity trackers, doing symptom surveys, and follow-up. And so we'll have those results probably also closer to the 1 to 2 year mark, because I think this one just got started as well. And then finally, recover sleep. Recover sleep is really to address some of the issues around sleep disturbances and also hypersomnia, so being excessively sleepy, and using a set of potential pharmaceutical interventions to address this. So a couple of different interventions to evaluate whether or not this can have an effect on reduction of the hypersomnia or the sleep disturbances that have happened with long COVID. And then finally, I just wanted to talk a little bit about opportunities for people in the community, both with and without long COVID, to be able to contribute ideas for potential clinical trials. And so we have an intervention information request form, and here you can submit a therapeutic for consideration in future trials. And it's really intended for use by all individuals, patients, caregivers, scientists, and then the information about how to get to this website is here. And here you can put in information, and then the NIH is planning on gathering all of this information and making decisions about ongoing clinical trials. In terms of the publications, so we don't currently have any publications from the clinical trials because, as I talked about, they're all currently either ongoing or just completed, but we anticipate that we will have them in the next several months to 1 to 2 years. And as these publications come in, they'll be posted on our study website, along with all of the publications that Dr. Goldman had just reviewed with you. So this is our study team. I just wanted to acknowledge that the study is really something that was a lot of work, and we've enjoyed working with all of the participants, and it's a huge team of us that work on this together. So thank you for participating. Thanks for listening to my talk, and I'm going to go ahead and turn it back to Dr. Heath now.
Thank you, Helen. And I've only got, I think, 3 slides to share, and one of them you've actually seen already. This is a slide that Dr. Goldman had put up, and I would just wanted to say a few words about it because it is the first, I guess, molecular-like diagnostic of long COVID. It's not perfect, but it does identify long COVID versus healthy by something like a factor of 2 more likely to have this. It's to give you a feeling for the nature of an assay like this. This analysis is done right now exactly in 1 lab in the world. It's basically, when you're looking at these counts here, you're actually counting copies of these spike antigens or N antigens or S1 antigens. But it's the kind of thing where, you know, for quite some time, we've been looking for any signal at all to try to have a molecular analysis of long COVID. And at least now it looks like maybe we might have one, even if it's, you know, I would say barely there. This has been analyzed with respect to the types of symptoms that individuals see. And I think here I showed this slide not just because it highlights that perhaps cardiopulmonary and neurologic and other symptoms may be more associated with these SARS-CoV-2 antigens, but also to emphasize why we do this national study. Because if you look, there's studies from the East Coast, 2 studies, the recover studies include a signal from up here in Seattle, as well as the Allen Institute. But also a 3rd location. And they're not all the same. And so one of the strong motivations in terms of the design of the recover study and the continuation of keeping the cohort engaged is the fact that we actually need to have such a diverse cohort if we want to begin understanding the nature of you know, I guess, molecular analysis and how that associates with the disease symptoms. But we really are just now beginning to do molecular analysis on these samples. And, you know, my strong guess is that these molecular analyses are going to be led by genetics. We initially were not going to do whole genome on the entire recover cohort, but now, you know, after a lot of lobbying by the scientists who are involved in recover, most of the patients will have their genome sequenced. And this is motivated by several factors. First, COVID-19 infection severity. So the acute disease, the disease that everybody caught before they had long COVID, is now the severity of that disease is now strongly associated with genetic risk. And there's a few references below, but there's several papers out there. Oftentimes, when you see a genetic risk, you see what are called high penetrant variants. And these are common gene mutations that perhaps are shared by 5% of the population. Much more people that have genetic risk are captured by what are called polygenic risk scores, which are thousands of gene mutations, each of which contribute a small amount to elevate risk of some symptom or some condition. One of the motivations of having such a large study is that we should be able to pull out both genetic risk from high penetrant genetic variants as well as polygenic risk. And polygenic risk scores, because of the availability of public databases over the past 2 to 3 years, have really matured quickly. And one way that we will look at this is, so let's say that we do identify a genetic risk for some long COVID condition, or maybe long COVID in general. You're going to have people with long COVID that do not have genetic risk, and you're going to have probably a larger number that have a high genetic risk. And this allows you to begin separating out nature from nurture. And it's the kind of thing that can allow us to begin to make sense of clinical labs or more molecular measurements. If you imagine right now, all we can do is compare patients that have long COVID, particular symptoms, with ones that don't. And that turns out has not been, as Dr. Goldman went through, has not been very informative. But being able to hopefully look at the differences between nature and nurture, genetic risk and environmental factors, will give us a much higher resolution. And in fact, that is beginning to happen in terms of interpreting the genetic risk for acute COVID. So more to come. We're in very early days of having this type of data, but we are excited to be starting to get it. I think we will start having a semi-regular series of these town halls with the folks you see here, and maybe some of the other folks from our other sites that are involved in the Recover program, to keep people updated with the clinical trials and the scientific findings that that are coming to light. So with that, we'll start having a Q&A. So the two people you haven't met yet are Helen Nguyen from UW. She's a coordinator in Dr. Chu's team, and Allie Duven from Swedish, who's a coordinator within Jason's team. So because we're just now meeting you, I'm going to direct some of the questions that we've gotten from the audience, and I'll start with Allie. So Allie, maybe you could talk a little bit about what patients can expect in terms of return of results.
For Recover, we have kind of two different kind of results that we can patients often ask about. The one set of results that you will get returned back to you often via MyChart are your clinical lab results. And so those are things, those are the lab results that you can review with your primary care providers and that you'll have access to via MyChart. The other set of results that people often ask about is their biospecimens. So those are the research labs that we collect alongside the clinical labs. And those are samples that get sent to Mayo Clinic. And so those set of results are not returned back to patients, often because they're being used for future research like we discussed tonight.
And I think also many folks are worried about the genetic information. We'll be collecting genetic information. Those will not be released back. There's several reasons for that. One of the primary reasons is that in order to make this a cost manageable program, the genetics are not being collected in a lab that would be sort of a clinical lab level genetics. They're more of a research level genetics. And so even if you got them back, there's nothing actionable that a physician would be allowed to act upon. And a second factor is, well, I guess that I think that's enough of why that's not done. It's just really a cost-effective approach. And so, Helen, Nguyen, maybe you could comment on, for those participants who have participated in some of the clinical trials, is there going to be an unblinding? Will they find out if they received the drug or placebo or what they received?
So for the 2 clinical trials that are ended or close to ended, Recover Neuro and Recover Vital, there are plans to unblind the patients who have participated in the studies. That will come out once the final study results have kind of been wrapped up, and it's expected to be in either spring or summer of this year. But your site, whatever your site that you're participating in, will have more information for you, and will be giving you official letters of those results as they come out to them.
Yeah.
Great, thank you.
Thanks, Helen. And just to clarify for some people, unblinding just means you find out what you received in the trial. So we've had a lot of questions about that, and good news is we get to know. Not all studies do that, but these Recover clinical trials will.
Okay, so for the next several questions, I'm just going to toss a question out there, and someone will physically raise their hand, and I will call on you. We were asked, is an elderly person more at risk to get long COVID, even if they're up to date with their COVID vaccines?
I can take that one, Jim.
Okay.
Yeah, so there are different risk factors for getting long COVID, and age is one of the risk factors that has been written about. There's been some very good community-based survey studies out of the United Kingdom and the UK, and these have actually shown different results. Some studies have shown that age does increase the risk for long COVID. Others have shown sort of that the middle-aged group is at the highest risk. So there's been some various different results there. But I think one takeaway is no age group is really completely spared from long COVID. It can occur in people of any age.
Okay, another question that we were asked, I'll paraphrase a little bit. What are we seeing in patients that are 5 years out in terms of the evolution of long COVID, patients who perhaps are having sporadic symptoms or not, or consistent symptoms? Yes, Helen.
I was going to say that it seems to vary a lot, that a lot of people seem to either have resolution of some of their symptoms, or they have flare-ups that happen and then they resolve and then they come back. And so it does seem to be person dependent, unfortunately. We have seen that in general, though, that as you've acquired long COVID later and later in the pandemic, does seem like it's less severe when you acquire it, and that's probably this effect of having the protection from the vaccines, and then therefore a less severe initial infection, and therefore less risk of severe long COVID. So.
Yeah, and there was a study, I think it's only in preliminary form, but some of the data was presented recently, which showed that it's not just important to be vaccinated, but having vaccinations that are up to date. So the protection And there's some caveats and et cetera with this, but the protection is largely you're protected 6 months post-vaccine. That's that and shorter times, but much longer times the protection is actually waned quite a bit.
Yeah,
Well,
that's correct. And that's part of the reason why you see the CDC has changed recommendations, especially for older adults and those who are immunocompromised, to be getting your COVID vaccine every 6 months to stay as up to date as possible.
Okay, here's a question. We'll probably need to define some of these symptoms, but I'll just ask a question with the acronyms. Are POTS and postprandial hypertension symptoms of long COVID? And if so, is there a treatment?
Yeah, I can take that one to start at least. Yeah, so POTS is postural orthostatic tachycardia syndrome. So it's a syndrome where you can have autonomic dysfunction or problems with how your body regulates the blood pressure and heart rate. And specifically, you get high heart rate without really having low blood pressure. And this can be very unnerving to people and, quite debilitating at times. So people with long COVID do have POTS. It's kind of an overlap syndrome. So this is, we don't know exactly how to cure it. There are different treatments for it, many different treatments. People have tried, salt loading or compression stockings, staying hydrated, drugs like midodrine sometimes. And these sort of approaches have been kind of worked out, before long COVID was around. And this is the sort of symptomatic management that is recommended when we can define a certain subtype. So these are things that you can talk with your primary care doctor about. If you're feeling symptoms of palpitations or fast heart rates when you're getting up or exercising.
Thank you. So there have been some requests coming across that they would love to have this recording available after this evening, and it will be. We will send notifications. I think we have people who registered with an email address. We will send notifications out and when it comes along. Here's a question. This is kind of a tough question. What reassurances are you able to give us who are losing hope that we won't be forgotten or left behind? I can take a shot at that. But I would appreciate other people chiming in here. I think this is something that we are pretty acutely aware of because, after the pandemic, there was obviously, a lot of COVID exhaustion around the country. And then this study has gone on for quite some time. One of the motivations for keeping the study going and even letting it go for another few years is oftentimes post-acute conditions. They evolve. Most patients or participants in the study will tell you that they're evolving, and that actually does happen. And sometimes you'll see more serious conditions involved, or you'll see resolution, or who knows what. But there's plenty of examples of chronic conditions that have a kinetic course that can take several years. And so we've been pushing very hard to continue this. Also, we've only just begun to do treatments, much less treatments that are informed by, say, genetic or mechanistic studies. Most of the treatments are really formed by things we sort of had on hand before the pandemic. So there is a very strong push to keep the study going so that patients aren't forgotten, and that this condition is not forgotten. And if someone else wants to add to that, I'll cede the floor.
Yeah, thanks, Jim. I'll add a little something here. I do think that Recover is evolving. It's being managed by a different group at NIH moving forward, at least the clinical trials part. So I think we're going to see some real changes here. I also hope a lot of the questions in the Q&A were about speed and trying to get answers quicker. I also hope that we move faster. I think it's important, and many people are suffering, and we need to move faster. That said, this is an extremely complicated disease that, as Jim mentioned, we're only beginning to scratch the surface of understanding the kind of the mechanisms or the biological underpinnings that cause it. So this is a really hard space to operate in, but there's a lot of energy being poured into it. And, I think that there's a huge community of researchers that are working on this and trying to understand it, develop treatments to alleviate the suffering. So I think, I can speak for this group, but, we are not leaving you behind. We're working hard as we can to try to figure this out.
Yeah, I mean, and I guess I'll say a word on the speed thing too. I know that many of the participants in the study are very frustrated by the fact that we're a few years in, and most of the studies so far are epidemiological, and so they're not going to necessarily lead to treatments. They're going to lead to more of a definition of the disease. We're just now starting to get some of the results back from some of the clinical trials that were probably not purpose designed therapies for COVID, but were therapies that were more or less on hand. This is like a big battleship. I don't know. I can't really, we should be moving faster. And it's been actually very frustrating for us as well that it hasn't moved faster. But I don't know what to say about that, but we're making that message known as clear as we possibly can. So here's a question. How can I best use the recovery resources with my care team?
I can start with this one. So again, this kind of goes back to the results that we have available from the Recover study are going to be the clinical labs. Oftentimes, at least for Swedish patients, if you're within the Swedish network, your provider is able to see them, the results of the study in Epic. And if you get most of your care outside of Swedish, feel free to reach out to our nurse practitioner, and we can or your study coordinator, and we're happy to help relay those results to the provider.
Yeah, and to add with that, a lot of our participants do also mention that they have a hard time getting their primary care doctor or other specialists to accept that they have long COVID or to even diagnose them with it. And so some people find it really helpful to have, in my chart, you're actually a part of the study as a patient. So your doctor can see that. And some people find it really helpful to point that out and to say like, hey, I'm in the study because I have these symptoms and I'm being researched on actively. And so that is also part of your medical records, and it kind of rounds out the picture of what you're going through.
Thank you. So we've had a couple of questions on nicotine patches, which seem to be something in, I guess, social media or something like that have popped up as being a treatment. So I think, Jason, you might be able to comment on that.
Yeah, sure, Jim. I could take a stab at it. I think, in response to this question, I did a little bit of reading and seeing what I could find about it. And the answer is not really much in the published literature about nicotine helping with long COVID. I think there was a very small study of 4 people that was published, and that got kind of picked up and sort of amplified in social media and chat groups, and people are experimenting with this. I don't know if this would work or not. I think is the bottom line. There's some, ideas about how it might work on inflammation, which is one of the theories about long COVID. So this would be something that would be good to test in a clinical trial. And I think, Helen, mentioned the NIH is soliciting ideas for clinical trials, and anyone's can give their input there, whether you're a patient or a health professional or a researcher. All are welcome to submit ideas there, and I did put the link to that in the Q&A. I hope you can see that, and If not, we'll try to send it out after the webinar.
I was just going to add that both in response to that and also about sort of the cadence of how things have been done in Recover. So I've done a lot of clinical trials in my life, as has Dr. Goldman. And a lot of the way that we do clinical trials is that we first need to understand what we're trying to fix. And we need to understand how to measure that improvement. And so for us to be able to do that effectively, we really need to fully and carefully design how we're going to do those measurements. And so what I sort of see as the first phase of Recover, these first couple of years, is really taking the data and saying, okay, this is what long COVID looks like. These are the different symptom clusters of long COVID. And so when we are designing a trial to be able to fix one certain set of symptoms, this is how we measure it. And so even though it seems like things have not happened and we don't have the results from the Paxlovid trial, and we don't have the results from the neuro trial, all of this pre-existing work that's happened in the last 2, 3, 4 years, really to understand how to even do the trial, that had to happen first. Because what you can't do is start a trial of something and not know how to measure its effect. And because those types of trials don't work. And then they give you answers that are uninterpretable. And then I think wastes a lot of people's time, and it's frustrating for the participants and for the researchers. So that's part of the reason. I don't think it justifies how slow that things have gone, but I just wanted to provide a little bit of background of why things happen in this order in this space. So.
Yeah, thank you. And I'll comment, maybe come back on a little more positive than I was before on that as well. So, I think as many people who are experiencing long COVID appreciate once you begin understanding what's going on in chronic disease in general, chronic disease has been the untreatable for many years, where it's difficult to get a diagnosis. You're told take 2 aspirin, get rest, etc. We are looking at long COVID as the opportunity to begin understanding chronic disease at a level that allows us to begin to treat it. And one of the reasons for, using caution or going thoughtfully forward, making sure we have the disease identified, defined, that we have a cohort, that we have longitudinal studies of patients, we understand what the clinical labs do or don't tell us, etc., is because that is what the opportunity here is. It's actually a spectacular opportunity to address a disease condition that has been virtually unaddressable in the past. And but it warrants moving carefully because it is very complicated. Everybody looks different in this disease to first order. Okay, so here's a question that was directed at me. I indicated that COVID-19 infection severity is associated with genetic risk, and antigen concentration may be associated with long COVID. In light of these observations, why do you think that infection severity is not associated with long COVID incidence? Well, there is actually an association.
There is an association. So people who have more severe initial disease are at higher risk of getting long COVID. But as time has progressed and people are getting less severe disease, they're at lower risk of long COVID. So there's probably both a genetic risk and then a disease risk. So
that's right. And I also suspect that some of the same genetic factors that are associated with acute disease may be associated with long COVID, but there are surely others. And your immunological memory is one factor that probably doesn't have a big impact on your initial acute response, but certainly is going to have an impact on long-term, and that can have genetic risk. There's all kinds of things. There was a question in here on autoimmunity. Well, that's an area where there are actually pretty good polygenic risk scores now. A survey of a much smaller cohort called the All of Us cohort identified about 1,000 people in that cohort that had long COVID that you could, particularly identify. And you could see that there was a little bit of increased long COVID with a higher autoimmune genetic risk. So I think those things It's still very early days, but those things are going to be resolved over the next few years. So let me just toss a couple more out here. Microbiome manipulation. Have any of y'all heard anything about how individual microbiome manipulation is relevant for long COVID treatment?
No, I also haven't heard about this. There's been a couple of studies that have looked at it, and I would say this is still very much in the theory realm, and we don't have enough information to start manipulating patients' microbiomes. That the standard there would be to try one of these types of manipulations in a trial. And that would probably be the safest. Manipulating the microbiome can have harmful effects too. So these are the things that we have to consider when when recommending treatments. It should have a strong evidence base behind it. And my reading of the microbiome literature is it's fairly confusing.
Yeah. But it's a rapidly evolving field. We have a lot of people working on that kind of thing here at ISB, but also all over the world. So it's also a story that, because we did take microbiome from, we took stool samples from many of the participants, and so that is a kind of a piece of information that will be folded into the studies over the next couple of years. So one question, and I think a few of us can answer this, is the RECOVER study over, or if it continues, what does it look like over the next few years?
So the RECOVER study is not over. 2 of the clinical trials are completed. The other trials are ongoing. And then the observational study where we follow people over time, that's continuing. We don't exactly know yet from NIH to what degree that will continue and who will continue, but the studies are ongoing. I think the NIH recognizes that this is really an important resource, and it's really, really important to continue to follow people past this year. So it is ongoing. We just don't think we have a lot of details quite yet. And I saw that there were also some questions about, what will happen with the new administration. That is also something that we do not know.
Yeah, I on the one hand, the new administration, well, not obviously very fond of vaccines, they do seem to care about chronic ailments. And so I think there's maybe a chance that they will be supportive of this. There's certainly a lot of congressional support for this. How the RECOVER program evolves, it'll phase 1 of it is slated to end this coming fall. And so there's many discussions right now, I'm involved in them, on what phase 2 is going to look like. It won't be as aggressive of a study because it'll be more focused on the clinical trials and on the analysis of the data that we have, but we will hopefully continue to monitor patients on a less frequent basis, but continue to monitor patients so that we can get an understanding of the kinetic trajectories of long COVID, what symptoms resolve or emerge over the next several years. I think that's actually a pretty good spot to wrap up. So we will do this again probably in 4 months. We're going to try to something like a trimester type of approach to keep everybody updated on how the study's going. I think results are coming out fast enough that that's a reasonable cadence. So keep on the lookout. We will post this, and we will reach out to you with this webinar location, and that you can share with your friends, and with the information on the RECOVER website that hopefully you can provide, give you some information for yourself or for your care team. And so thank you all very much for attending. It was a pleasure to be able to do this evening.
Yeah, thanks to all the participants. I'm sorry we couldn't get to all the questions. Thanks for the robust discussion in the Q&A, and we can try to answer some of these starting at the next town hall. So thanks again for your participation in these studies.
Thank you, and have a good night.